Good evening, students. In today's class, we are going to learn about biochemistry of aging. In today's class, we are going to learn the different theories about aging, what are the biochemical significant changes occurred in the body during aging, and is it possible to reverse the aging? Aging is a natural phenomenon. The lifespan of the individual increases over a period of time. In the medieval period, it was around 25 to 30 years, and it is increased to 40 years by 20th century, and now it is increased to 67. In developed country, the lifespan of human being is 80, and in India, more than 10% of the people are having lifespan more than 60 years. As we get older, all the physiological process decline. This declination in the physiological process will differ between individual, and the reason is because of the environmental factor and genetic factor. And those people with diabetes mellitus, hypertension, dyslipidemia, obesity and reduced physical activity has been found to have early onset of aging. There are several theories which explains about aging. We are going to see about each of this theory one by one. First theory is wear and tear theory of aging. According to this theory, cells sustain damage over a period of time due to injuries, disease and environmental factors like pollution. Surprisingly, this theory also states that even the substance which is taken by us routinely like water, oxygen, sunlight also causes damage to our cell. So let's see how water and oxygen can cause damage to our cell. So let's start with water. So water is a weak nucleophile and it occasionally is going to replace the amino group which is present in the side chain of the amino acid and nucleotides. So by replacing the amino group present in the side chain of the amino acid and nucleotide, it is going to convert this amino acid and nucleotide to its corresponding amino acid and nucleotides. That will result in change in the sequence of the amino acid in the protein and sequence of the nucleotides in the DNA. So that is going to damage the DNA and protein, that will result in aging. So let's see what are the amino acid and nucleotides which is going to be altered. So I told you the amino group present in the side chain of the amino acid will be removed. So which are the amino acid which has amino group present in the side chain? All the amide containing amino acid, right? So which are the amide containing amino acid? Asparagine and glutamine. So asparagine and glutamine are the amino acid which is going to be involved in hydrolytic reaction by water. So by hydrolytic reaction, we are going to remove the amino group present in the side chain of the amide containing amino acid. So it will get converted to form its corresponding acidic amino acid. That is asparagine will get converted to form aspartic acid. Glutamine will get converted to form glutamic acid. That is glutamate. So this is how amide containing amino acid will get converted to form its corresponding acidic amino acid. So we are changing the amino acid instead of asparagine or glutamine in that particular protein amino acid sequence, we are converting into an acidic amino acid. So the sequence is affected. So the protein structure is affected. That will result in damage to the cell. That will result in aging. Next, coming to nucleotides. By hydrolytic reaction by water, which are the nucleotide is affected? The nucleotide which is affected are cytosine and adenine. In case of cytosine, we are going to remove the amino group, so it will get converted to form uracil. In case of adenine, here when I add water, this adenine will split to form ribose 5-phosphate and its corresponding nitrogenous base. So this is how nucleotide will get converted. So the DNA structure is affected. So that will result in aging. Next, let's see how oxygen can cause damage to the cell. So oxygen can cause damage to the cell by getting converted to form reactive oxygen species. In the previous class on free radical, you might have learned from oxygen how different reactive oxygen species are produced, like superoxide, hydrogen peroxide, hydroxyl radical, lipid peroxide and thiol radical. So all this free radical will damage the nuclei, DNA as well as the protein, which result in aging. This is how oxygen, by getting converted to form reactive oxygen species as free radical, is going to damage the DNA and protein, result in aging. Next, coming to the second theory, that is mutation of mitochondrial DNA. So in the previous slide, we had learned free radical can be produced from oxygen and that will damage the human DNA. So apart from this, it will also damage the mitochondrial DNA. So the free radical can damage the mitochondrial DNA. So the protein which is produced from the mitochondrial DNA will be reduced. That will result in reduction in the activity of electron transport chain. That is, electron transport chain will not occur properly. So ATP synthesis will be reduced. That will result in cell damage and aging. The third theory is reduced activity of telomerase. So what is meant by telomerase? In replication, you might have learned in the first stage of replication, the double-stranded DNA will split. It will split to form single-stranded DNA. So following this, at the 3' end of the template strand, that is at the 3' end of the template strand, a primer will be added. And following this, DNA polymerase will get attached to the single-stranded DNA, and it will start producing the new strand from 5' end to the 3' end. And once it has produced the new strand, it will get separated. Now the primer will be removed. So now if you see here, following replication, there is a shortening of the DNA, right? There is a shortening of the DNA because the primer is removed. There we don't have any nucleotides. So to prevent this, if this continue, after every replication cycle, the DNA will get shortened. So we don't want that to happen. So what we are going to do, we are going to add multiple tandem repeats of nucleotide at both the end. So we are going to add multiple tandem repeats of nucleotide at both the end. So this green color is the multiple tandem repeats, which is present both the end of the chromosome. So by adding this, after each cycle of replication, the shortening is prevented. So this multiple tandem repeat is called as telomere. This tandem repeat is called as telomere. So telomere is going to protect the DNA during replication. So even the telomere will get shortened after each replication, which is prevented by telomerase. So telomerase will prevent the shortening of the DNA. That is, it will start adding new nucleotide in the telomerase center. So telomere is a tandem repeat of AGGG-TT sequence. So telomere will get shortened after each cycle of replication, which is prevented by telomerase. So as we get older, this telomerase activity will be declined. It will reduce. So because of that, shortening of DNA will occur. That will result in cellular damage, cell death, and aging. So this is how aging will occur due to reduced activity of telomerase. The next theory of aging is mutation theory. So in this theory, following replication, there will be some error in the newly produced double-stranded DNA, which is corrected by proofreading. And also every day and over period of time, there will be minimal damage to the DNA due to the environmental factors. So which is corrected by DNA repair. Even though our system repairs this damage, it is not 100% perfect. So there will be minimal damage, and this minimal damage which get accumulated over period of time, which is going to damage your cell, which result in aging. This is called as mutation theory of aging. Next theory of aging is error catastrophic theory. So according to this theory, there is something called as aging gene. So this aging gene will cause error in the amino acid sequence of key enzymes. So these enzymes are required for production of several proteins. So these proteins also will be produced in defective because of this aging gene, which has an impact over the key enzyme with abnormal amino acid sequence. So because of this abnormal protein, their functions also will be affected, and these proteins are involved in regulating genes which are going to be involved in the production of other enzyme involved in protein synthesis. So it is going to become a vicious cycle which is going to affect the cell, that will result in aging. The last theory of aging is pacemaker theory. So according to this theory, aging occurs due to decline in immunological system, like decrease in the activity of T cell and autoantibody against our protein, which result in damage to the protein and nuclei, which result in aging. So these are the different theory of aging. Next, coming to the significant changes occur in the body during aging. As we get older, there will be sarcopenia, which result in loss of muscle mass. There will be osteopenia, which result in osteoporosis. There will be oxidative stress, which result in lipid peroxidation. There will be decreased antioxidant, increased insulin resistance, glycation of protein, which result in diabetes mellitus and coronary artery disease. And there will be loss of subcutaneous fat, which result in wrinkling, and loss of elasticity of the collagen, which result in stiffness of joint and tissue. So these are the biochemical changes which occur during aging. Coming to the last part of the session, whether aging is reversible or not. Let's see. So is it possible to reverse the aging in the future? Let's see. So to understand this, you should know what is meant by CRISPR. CRISPR stands for C for clustered, regularly interspaced, short palindromic repeats. So it's like a molecular scissor. Let's say a bacteria is having a CRISPR and that bacteria is infected with a virus. And that bacteria will use the CRISPR to cut the DNA of the virus, and it will store one of the fragment of the DNA of the virus in its DNA. So that fragment of the DNA of the virus is called a spacer. Now it is called a spacer. When same virus infect the bacteria again, this bacteria can able to recognize that virus using that fragment of the DNA which is already stored, and it can able to destroy that particular virus. So how we are going to use this in reversing the aging? If you see, normally as we get older, the gene which is required for normal health will be down regulated, and the gene which is going to promote the disease will be overexpressed. The CRISPR technology has the capacity to make changes in the specific part of the DNA, so this can be altered. That will result in prolongation of the that is delaying in the aging. So that's all about biochemistry of aging. So in biochemistry of aging, we had seen about different theories of aging and what are the biochemical significant changes occurred in the body during aging, and about CRISPR. Thank you.